Okay, so we're going to talk about protein binding and function. And we are starting out, we're talking about globular proteins now. So these are proteins that have at least tertiary structure, where we have that polypeptide sequence, primary structure, and then we have either beta sheet or alpha helix. And then we have tertiary structure, which is causing the shape of this protein, and that tertiary structure is being held together by mainly non-covalent interactions of the R groups. So the first type of protein binding that we're going to talk about are in antibodies. So let's talk about what an antibody is. So an antibody is made by our immune system, and it binds what's called an antigen. And you'll see antibodies abbreviated as AB and antigens abbreviated as AG. So there's 5 major classes of antibodies, and you're not going to be responsible for knowing each of these, but these are produced in response to some non-self organism or molecule, which is what the antigen would be. So this antibody-antigen binding is important. It is one of the early signals of our immune system that something is wrong and that we need to fight some sort of infection or something that's not there. So that shouldn't be there. So the 5 classes of antibodies, they're all given an IG at the beginning. IG stands for immunoglobulin. Immunoglobulin is another word for antibody. And again, like most science words, it tells you right in the name what it is. So immuno means this is part of our immune system. Globulin means this is a globular protein. So there's 5 classes. There's IgM, IgG, IgA, IgE, and IgD. And they all are different, serve different purposes in our immune system. There's a chart about them in your PowerPoint. You do not need to know all of the different types. We are going to focus on the structure of the IgG molecule. So the IgG molecule is made up of a few different parts. So it has what's called variable heavy chains and then variable light chains, and constant heavy and constant light. So there's heavy chains, variable, and constant. And then there's light chains, also variable and constant. And we're going to see the edge of these variable chains are where we're going to bind the antigen. So all IgG molecules are going to have the same constant region, both heavy and light, and then they will have specific variable regions specific to whatever that antigen is. So there are better pictures of this drawn in your PowerPoint lecture and in your textbook, but I'm just going to draw them too. So it kind of looks like a Y with 2 extra pieces on the outside. So what we have here, where this is where the binding would happen, this is where the antibody would bind the antigen. And these chains here in the middle, these are heavy chains. And then this chain here and this chain here, these are the light chains. So at the tips, so for example here, This would be a heavy chain, and here is the variable region. So this we would call variable heavy. This is the light chain, and this is the variable region because it's where it binds the antigen. This is variable light. So variable heavy, variable light. And then the constant region, same thing. So everything else here is constant. So this is going to be constant heavy, constant heavy, constant heavy, constant heavy. This would be constant light. So it's made up of these different regions, and this is just showing this is the constant part of the IgG molecule all down here, and then this is the variable part of the IgG molecule, which is specific to whatever antigen it's binding. So this is a protein, so it's made up of amino acids. So the R groups that are here in this variable region are going to bind non-covalently to the R groups of whatever that antigen is. So it's usually a protein in the antigen that our antibody is recognizing, and then it's non-covalent protein-protein interactions between the antibody, which is our protein, and the antigen, which is some foreign protein. So we'll talk about this more when we get into the, well, maybe we'll talk about it now. So there's a lot of immunology stuff in the PowerPoint of this lecture, and you're not responsible for all of that. I include it just for you to see the bigger picture. What we're interested in are these protein-protein interactions between the antibody and the antigen. So let's talk a little bit about some antibody applications and techniques. So there are many, and we're only going to talk about 3 today. One is called the ELISA. One is called a Western blot. And the third that we'll talk about is microscopy, specifically immunofluorescence. So because these antibody antigen, this binding here is highly specific, this antibody isn't going to bind to anything else but its one specific antigen. So we can exploit this in science when we are using these for tests in a lab or many types of research. We can create antibodies to a specific protein that we want to learn more about. And once an antibody has been created to that protein of interest, it's kind of acting like an antigen, but this is some sort of situation in a lab where we're trying to learn more about a specific protein, we can join all kinds of things to this antibody to then learn more about that protein. Not in this section, but another thing that is really important that we're able to do with antibodies is that we are able to specifically deliver drugs to important locations by conjugating the drug actually to this antibody, and then the antibody will go around and find whatever protein it's supposed to find and deliver that drug specifically to that protein. So this is important. This is something that happens. We use a lot of what's called immunochemistry and techniques like this in cancer, where we're trying to deliver chemotherapy specifically to the cancer cells, because all chemotherapy is poison that is trying to kill more of the cancer cells than the good cells. So when you have this targeted approach and you're able to target some sort of cancer protein, a protein that's only going to be found on the surface of cancer cells, you could make an antibody to that protein that only the cancer cells have, and then link the drug to that antibody, and then you're essentially delivering the poison chemotherapy straight to the cancer cells instead of having it affect the whole system. So we use antibodies a lot in medicine. And then we'll talk about these 3 techniques too. So the first one is called an ELISA. And what an ELISA stands for is it's an enzyme-linked immunosorbent assay. ELISA, enzyme-linked immunosorbent assay. So what you're doing is you're linking an enzyme to an antibody. Anytime you see the word immuno, we're using immunology, which is antibodies. This is what happens when you're sick and you go to the doctor and they said, we're going to do a rapid strep test. We're going to see if you have strep throat. And they're going to swab your throat, and then they're going to go away. In 15 minutes, they'll come back and say, you have strep throat or you don't have strep throat. What they're doing when they're testing that swab that has swabbed your throat is they're looking for the protein on this strep bacteria that would be causing strep throat. And this immunosorbent assay usually has some sort of what we call a fluorophore linked to it, which is going to cause some sort of color change. So what they're doing when they're doing this rapid strep test is they swab your throat, and then they go put your sample in another sample that has strep throat antibodies in it, strep bacteria, antigen. And if your body has strep antibodies, that means your body is fighting that strep bacteria right now. And they have linked something that's going to cause a color change. So they're going to put this in a little well plate back in the doctor's office. And if it changes color, that means that your body had produced those strep bacterial antibodies against that protein on that strep bacteria. And that means you have strep throat and you need an antibiotic. This is also something that is being worked on right now with the coronavirus. Now, this is not the swab test for the coronavirus. The coronavirus is an RNA virus. And when they're swabbing the back of your nasopharynx for the coronavirus test, what they're doing then is they're using something called PCR, which is then going to amplify all of the RNA to see if you have that coronavirus RNA. What we eventually want is a blood test that would do this, a serology test, to see if we have in our bloodstreams already the antibodies to coronavirus. So there's a lot of talk about people saying, well, I was really sick in January and I might have already had coronavirus. This would be the way that they would test to see if you did. This would be a blood test. The strep throat test is a swab. The swab for coronavirus is not this. It's looking at RNA. But this is something that probably a lot of us will have, I'm guessing, in the next year or so. I wouldn't be surprised if a lot of us had blood tests to see if we actually had coronavirus earlier this year. And potentially what that means is if you've had it, then you would have some immunity to it because your body has already seen it, and you would have some of those antibodies. This is all just my best guess at what is going to happen, and we'll have to see what happens this fall. But that is an ELISA. So we're able to use antibodies, and the way that our antibodies produced by our body have such a high specific binding with an antigen, which is anything that an antibody binds. The antigen doesn't have to be a pathogen. Pathogen means like a bacteria or a virus or something external that causes a disease. So that's an ELISA. So we use this for diagnostic tests. This is also similar to how a pregnancy test works. Pregnancy tests are looking for levels of hormones and proteins in urine. Same thing, there's a fluorophore conjugated there that causes a color change if that protein is there, indicating pregnancy. A Western blot is another tool that we can use using antibodies. We use an antibody to visualize a single band of a specific protein in something called an SDS page, which is a gel. And there are pictures of this in your textbook and PowerPoint. I'm not going to draw it, but we're using antibodies to detect a specific protein. And this would be experimentally. If you were seeing how a specific experiment affected the expression of your protein, and then you could use a Western blot to see if your protein expression went up or down, something like that. But this is another way that we can use antibodies in different techniques. The third one is microscopy. This is called immunofluorescence. Similar to linking this fluorophore in this enzyme-linked immunosorbent assay, but now we're looking at things inside cells under a microscope. So we can conjugate these immunofluorescent molecules to these fluorescent dyes to different parts of the cell, and then we can visualize them under a microscope. Whenever you're looking at pictures of cells under a microscope and you see all these different structures and they're red and blue and purple and green, that's not the color of the actual structures inside the cell. Those structures have been specifically dyed using immunofluorescence, where we're taking a protein that we know binds to DNA, and that's going to turn the DNA blue. So then anywhere you see blue inside the cell, you know that is DNA. Or we're taking an antibody that has red attached to it